So some of you who attended 2 years ago may be asking yourselves the question, does Alex only own one suit? I own 2 suits. I own 2, but the other one's on campus. So welcome to day 2 of the Phage Canada Virtual Symposia. I'm very excited about today. Yesterday was really a showcase that we couldn't have done any other way. I don't think we could have ever created an in-person event that was able to get undergraduate researchers from so much of the country sharing their research. I think it was really playing to the strengths of this format, and I had a lot of fun. Today is a bit more traditional and a bit more in the way that we have done things 2 years ago, to focus on trainees, graduate and postgraduate research. And tomorrow is again something a little bit different with career opportunities in phage research. Now, I first want to take a moment to thank our sponsors with a particular emphasis on the Canadian Society of Microbiologists. So they have given everything that we need to be able to run this through the 4 Waves platform, to do this and make it available to all of you free of cost. And so we're incredibly grateful to them and their initiatives in sponsoring efforts like these. But we also have 3 phage-based companies who have provided sponsorship for awards for talks, including some of those yesterday and some of those today. So we're very excited about that support and that continued support in the case of 2 of these companies, that I think is really helping us build this network. For other acknowledgements, I am standing here in front of the camera, sitting here in front of the camera, but the vast majority of the work to bring this together has been from this organizing committee, all of whom are trainees. And I think that's really what I want to make a little point about. This, in its past iteration and its current iteration, has been entirely focused on trainees. And so you might be asking, well, why do we have a keynote lecture? Why would we break from that format when everything has been organized by trainees? All the presentations have been by trainees. Andrew is many things, but I don't know if he counts himself as a trainee anymore. And you know, last time we presented this, I, half jokingly said that I started this whole thing because of a CBC article that said there were only 6 phage researchers in Canada. Partly true, but partly an exaggeration. But another thing that has also made its way into my grant proposals is this idea that there's a big disconnect between our past, because there were a generation of scientists that weren't trained on phages, because there was a loss of funding, because there was a long period where new phage labs weren't created. There's a gap in our history. And I think it's really important to make sure that we reach back and connect to some of what happened before, and not lose that. And so we have an opportunity to fix that today. I also, I won't spend a great deal of time introducing Andrew. I think he's going to go a little bit over his academic lineage and his phage history. My first introduction to Andrew did not look at all like this, but looked like this. And I think many of you may as well be dependent on this Bacteriophages book, sort of the gold standard for bringing yourself up to speed on the techniques around. But Andrew does have the claim to being the original Phage Canada, as we found out when we tried to register the email address phagecanada@gmail.com. Someone had beat us to it. So without further ado, I'm going to hand the reins over to Andrew. I'm going to stop sharing my screen and let you share.
Okay, great. Thanks so much, Alex. Okay, my assistant is helping me. That's the one there. And then we're going to hide those guys. Okay. This definitely takes 2 people to run it from this part of the thing. So I want to talk about the history of phage research in Canada, and I'm going to start off with sort of putting things in perspective. So you need to view the next few slides with respect to yourself. Okay, so everyone's got a family tree. So here's me down here, my parents, and my mother's relative 1,000 years ago. And through one branch of my family, I've got my son and daughter. Through another branch of the family, I have my, let's go, never mind. We have the family dog. Okay, all of you also as graduate students and faculty members have an academic tree. So here's me with my supervisor, Tony Warren, at the University of British Columbia when I was a graduate student and was younger and slimmer. We can trace back Tony's scientific parentage to his scientific father and to his scientific grandfather. And you can also trace back my lineage as to my scientific children, Ken Gerald, who was my first graduate student, and Catherine Paul, who was my last graduate student. The horrifying factor is now Ken has retired, which makes me feel thoroughly ancient. Okay, you can also look at, I'm going to get this little thing out of the way. There we go. You can also look at your discipline tree. So in the case of Tony Warren, his scientific father, Joe Nealons, worked on iron metabolism. And when he came to UBC as a young faculty member, he also worked on iron metabolism. I did. The project didn't work out, and he then switched with me to working on bacteriophages. And when I left, he continued a bit on that, and then he ended his career working on cellulases. And my first and last graduate students worked on archaeal motility and environmental microbiology. So the real problem that we saw with people of certainly my generation was the lack of discipline continuity. And really one would like to see, all of us working on bacteriophages, and this may happen. Okay, the problems really have been that the scientific funding agencies really haven't supported many phage scientists throughout their careers. And as a result, many of the really famous ones, some of whom you will hopefully recognize their names as we get to them, in fact, left phage research for better funded areas of research. Indeed, NRC, NSERC cut the infrastructure funding in 2012, which meant that the Felix Durrell reference collection was affected, and the cost of buying a phage is now, I think, something like $400 when it used to be about $50. And in addition, I had set up national and international collaborative efforts on phage projects for 5 years, and then I thoroughly got sick of it. None of them were funded, even though Canada said that it was interested in dealing with alternatives to antibiotics. So I want to talk about the history of what's gone on in Canada. And of course, the first question people are going to ask is, am I mentioned? And the first things I managed to get are, if you've died, maybe. If you're an emeritus prof or emeritus scientist, probably. And if you're a senior faculty member. So Alex, unfortunately, won't be mentioned. Not this time round, but maybe next time. Okay, so one night recently when I couldn't sleep, I decided to look up the literature on phages and find out what the first publication was by someone on phage in Canada. And the first person to work on this is E.G.D. Murray, the father of Bob Murray, who just died this spring at the age of 102 and taught at Western University. He was, in fact, the editor of the Canadian Medical Journal, Medical Association Journal, and wrote a very interesting article on the phage phenomena. This is before people really knew what phages were all about, and it's well worth reading it. 4 years later, a surgeon at the Montreal General Hospital used phages to treat a Staphylococcus aureus phage infection of someone's neck. It's worth reading because it's ghastly, but it worked. So that's that. So Canada was involved in phage research way well before you even thought about, mostly. Okay. So, but if you look for papers on phages, you're going to find from the 40s to the 70s that the major area for phage research was in diagnostics, in phage typing. And this really is illustrated nicely in this central part of the things where we have a lawn of bacteria upon which are spotted a number of different phages. And the lysis patterns, you've got complete lysis here and no lysis here. And in fact, to do phage typing, it actually takes quite a bit of training to be able to read these results in an appropriate manner. But we have a number of individuals. We have Roger Compter at the University of Montreal, worked on Staphylococcus. We've got Eden Bromfield, who worked on a phage typing system for Cynorhizobium. We've got two scientists, one in Alberta at a hospital and the other in McGill, who worked on Mycobacterium phage typing, and Hermie Lior from the National Enteric Reference Center, which no longer exists in Ottawa, on Salmonella. In fact, this is a picture of the last Salmonella phage typer who worked at the National Microbiology Lab in Guelph until about 5 years ago, when she retired and they retired the typing position. So now all strains, rather than being phage typed, are sequenced. One big area where phage have become important and has led into taxonomy is electron microscopy. And the big names there were David Bradley and Hans Ackermann. You'll notice that their names are in red, and wherever someone's name is in red, I have created a phage taxon. It could be a genus, could be a subfamily or a family in the case of Hans Ackermann, which are now recognized. Okay, so we talk about David Bradley. Everyone would have heard the Bradley classification of phages, which is a letter, usually sometimes followed by a number. So a C3 type virus is a potovirus with an elongated head. He also employed acridine orange staining, which he could figure out what sort of nucleic acids the phages contained. The specific, second sort of thing he was famed for was working on conjugal plasmid-specific phages. So these aren't bacteria-specific phages, these are plasmid-specific phages. And he came up with the pilus retraction theory, which has recently been proved to be correct. And since we're talking about single-stranded RNA phages, it's worth mentioning Bill Poranchuk from the University of Alberta, who spent his career working on how R17 infects E. coli. Hans Ackermann, 15 years the chair of the Bacterial Viruses Subcommittee of ICTV. He's the person who's singularly responsible for creating the order Cardioviralis and the three families which belong in that. I should mention right now that, in fact, those four taxonomic entities are being discontinued. So please keep an eye on ICTV for the latest taxonomy as it applies. So we now have an Ackermann viridae family. David Bradley has only got Bradley virus. He founded the Felix Thorel Reference Center. He observed over 6,000 phages by electron microscopy. And the wonderful thing about him is you could show him an electron micrograph and he could say, I've seen something like that. And he would immediately dig up the phage he saw. He was, without a doubt, the collaborator and the critic extraordinaire. To give you a story about this, I received a couple of electron micrographs from a scientist in Canada. And the person said, could you please have a look at these pictures and tell me what you think? And I said, well, you're sending them to the wrong person. You should send them to Hans. But please beware that if, in fact, he doesn't like them, he's going to write you a letter which will explode your computer, and it will suggest that you be sterilized and otherwise done away with. So the person said, well, I don't really like the idea of that. Would you please send him the pictures? So I sent him the pictures and I prepared myself for how I would defuse the letters I got back from Hans, but in fact, he loved them, which was great. Dr. Komme is because he was a grammarian extraordinaire as well, which comes down to me. I'm responsible with Rob Levine for coming up with this sort of name for phages. So we have the name of the phage, which is Tweedledum, and we have this prefix. The prefix has VB for virus or bacteria, and then something which tells you about the host and the morphotype. So this is an E. coli phage, which has a myovirus type of structure. I've been involved over the last 10 years or so when we moved from morphology-based, in other words, Hans Ackermann's approach, to a proteome and a total genome approach for macrobial taxonomy. In my spare time, which I've got lots of in theory, I've produced some 600 taxonomy proposals, which have produced several new families. And to keep my wits in, I offer phage annotation workshops. Okay, so if we take a look at phage in Canada, the big place where there was a lot of phage workers at the same time is the University of Toronto. And the two big names are Marvin Gold and Louis Siminovich. He died, I think, last year at least 100 years of age. He actually Louis produced this guy who continued working on phage. And Marvin produced Alan Davidson and Karen Maxwell. We're going to be hearing from one of Karen's students. They have been working on the morphogenesis and also lysogeny, and in case of Karen, also resistance mechanisms. So really a famous group of scientists assembled at the University of Toronto. Many of them went on to other areas. In Louis Siminovich's case, he went on to human genomics. Okay, continue with the temporary phages. Sidney Hayes is semi-retired from the University of Saskatchewan, worked on regulatory mechanisms. We had two people in Canada working on mu-like phages. George Draconis from the University of Calgary, who worked on mu before moving over to working on Lyme disease, and Michael DeBow from McGill, who was a really weird and wonderful fellow, and unfortunately died last year in Paris. He arrived at conservative Queen's University many years ago, dressed entirely in black with a white sheepskin wool vest on, and essentially blew the minds of all the conservative folks at Queen's. And lastly, we've got Barbara Funnell, again from the University of Toronto, who works on plasmid prophage replication. Now, most of the phages that we've talked about, lambda and mu, integrate into the chromosome. In the case of phage P1 and Pseudomonas phage F116, they replicate as plasmids. And she's one of the few people who are actually working on plasmid replication. Now, I didn't know where to put these folks, so I created a title called Biochemistry. And they're all linked by the fact that they all worked on phages which have modified bases in them. So the first person, the most famous person, is probably Jerry Wyatt from Queen's University. Before he arrived at Queen's, he is the person who identified hydroxymethylcytosine and glycosylated hydroxymethylcytosine in the DNA of the T even-like phages. Tony Warren worked on modified nucleotide biosynthesis in a Delphtia phage, which I in fact isolated. And Iwo, or François Takahashi from McMaster, worked on a bacillus phage, which has uracil in its DNA, not thymine. We've also got a couple of well-known and well-respected scientists who have worked on T4 DNA replication, and in the case of Paul Sadowski, also on T7. Francois has a genus named after himself. The other folks don't yet. In phage therapy, we've got people who met the criteria of age and brilliance. Jonathan Dennis from Alberta, who works on phage therapy against Burkholderia and Stenotrophomonas, and who has developed a variety of alternative models for phage therapy. So you don't need to do animal work or animals who have backbone work. You can work on other systems. And he's developed an aerosol system for delivering phage to the lungs, because these are organisms which cause cystic fibrosis. And Louis-Charles Fortier is working on Clostridioides difficile phages, or Clostridium difficile phages. Okay. Now, if one looks at the totality of work which has been done on phage research, it's predominantly in the area of agriculture that one finds the greatest number of scientists. And the first one that one can find is Gord Greer, working in agriculture and Agri-Food Canada. he was interested in the use of cold-tolerant phages, which would infect Pseudomonas strains infecting meat. And interestingly enough, I suddenly recalled as I was working on this, that Gord Greer was the first graduate student on whose committee I worked when I came to Queen's University, and unfortunately died relatively young. So Agriculture and Agri-Food Canada and the University of Guelph are the big areas of this. So if you ever get a chance to meet Parvis Sabor, please go and chat with him. You will never believe that he's only about 84. His life history really adds up to about 120 years. He was interested in dealing with mastitis in cattle, an expensive area for reduction of milk production. And he was also interested in encapsulation. So these people here are really interested in applied aspects and have illustrated some of the problems that you associate with applied aspects of this research. I mean, how do you introduce phage into the teats of cattle? And what do you do about the fact that, in fact, milk proteins inactivate one of the most active staph phages, staph phage K? Antoinette works on Erwinia phages and plant pathogens and is interested in using them to spray orchards. But if you spray orchards, what do you do about the rain? What do you do about UV inactivation? And we have Michael Hynes, the father of a moderator, who works on rhizobium phages, again, an applied area. These three individuals from Agriculture and Agri-Food Canada, University of Lethbridge, and the Public Health Agency of Canada have worked on and produced a number of manuscripts on the use of phages to reduce the carriage of E. coli 0157 in feedlot cattle. Because if you can reduce the carriage of this bacterium by cattle, even by, say 25%, you will have a huge impact on the possibilities of foodborne illness and a huge cost associated with that. They were also responsible for finding that when they conducted their phage trials, they actually put in one phage and they got two phages out. So when you're working with animal systems or environmental systems, things don't work quite as neatly as they do in the lab. And Roger Johnson is renowned because he worked out a number of techniques for enumerating phages. University of Guelph, which I'm associated, had a number of scientists who working on phage. Mansell Griffith, isolation of phages against a wide variety of foodborne pathogens, including Listeria, has isolated bioluminescent phages and worked on bioabsorbance. Carlton Gile, who's actually these two have now retired, worked on E. coli 0149 specific phages, and Keith Warren is still continuing phage work at the university on the control of human pathogens on plants. And of course, one can't forget the agricultural phage person, Sylvain Monod, working on lactic acid bacteria, the Caspar crisp system. He's been the curator of the Felix Sorel collection of phages since 2003, and he's the Canada Research Chair on Bacteriophages since 2011. And lastly, we have Aquatic. And Curtis Suttle definitely, Mr. Environmental virusphere person to deal with. And I put in here Eddie Ishiguro, who is now retired from the University of Victoria, who worked on Aeromonas phage receptors. So in my concluding thoughts are, we really mustn't forget our scientific heritage, and we should do everything possible to, in fact, create some sort of way of preserving information about the people who have worked on phages, and also people who worked in your individual departments. And in 10 years' time, which I hope to still be here, I want to see many, many more names of phage scientists and longer discipline trees. And so, I'm sure that this will definitely occur. in conclusion, I'd like to thank NRC and NSERC who funded me through my entire career working on phage, and especially Alan Davidson, Sidney Hayes, Michael Hynes, and Sylvain Monod, who in fact went through this presentation and were able to add very useful information to it. At which point I will say thank you so much for listening, and if there are any questions, I will do my best to answer. So I guess.
Excellent. Thank you very much. I will relay the questions